Well, good evening to everybody. Thank you for joining us to talk about the TAA therapy. This is put on by the Musella Foundation. Al Musella and Vanessa from the Musella Foundation are joining us and helping to or presenting this entire session. I am Keith Deserick. I'm also joined by Rachel, John, who is our research support manager. And we're here to talk with Dr. Hwang with regards to multi-targeted tumor-associated antigen T-cell therapy. This has been very much in the news. I know that many people have seen this and wanted to learn a little bit more. So we wanted to reach out to Dr. Hwang and learn a little bit about that. Before we go ahead and talk a little bit about the therapy, I want to introduce him. Dr. Hwang leads the Division of Oncology at Children's National Hospital, where he serves as the Willie Hughes Professor of Immunology and Neuro-Oncology. He is passionate about finding new cures for children with brain cancer that can give hope to families and has risen to prominence in leading cutting-edge clinical trials, especially focused on brain cancer. Working with a large team across the globe, including patient advocates, foundations, scientists, and other physicians, Dr. Hwang fights alongside patients to provide treatment for all children with cancer. I know that personally, I know him as an innovative leader in this field who tends to come up with those strategies that a lot of times we may overlook, we may miss, and does so with a passion that lasts years and years. It's a very strong determination, and I'm glad that he is part of this field. So, with that introduction, Dr. Hwang, do you want to tell us a little bit about the TAA therapy?
Yeah, thank you, Keith, and everybody else, Al, for inviting me to talk about it. It's obviously, Keith, as you talked about, it has had a lot of visibility for some good reasons. I thought maybe what I do is just start sharing. It's a little bit complicated. It's also kind of straightforward in some ways. So I thought I'd sort of share and just kind of move through a little bit about what this therapy really is. So this was the Remind trial. You can see that we kind of cobbled it out of a more complicated title of what it happens. And I obviously want to start with acknowledgments. I know these often come at the end of any talk, but like anything else, everything does take a really passionate village. And so we have this amazing team at Children's and also a lot of the people in the community like yourselves who were really important components to make this trial even possible, much less successful at all. And at the very end of it, it's all about patients, their families, their communities. That's exactly as Keith was talking about. Unfortunately, I think too many of us are actually members of both the families and patients, as well as the scientists trying to figure it out. We already know that in general, this was a trial, by the way, for that encompassed diffuse midline glioma, especially DIPG, but actually also included a bunch of other kinds of aggressive pediatric brain cancers. They kind of sit in the same bucket. If you're newly diagnosed, unfortunately, as you guys know with DIPG, or if your brain cancer has come back, then our ability to cure those becomes really difficult. And so we are really aiming to try to help all those children. We know that the kitchen sink is often thrown at children and their families, and those kitchen sinks come with pretty significant and toxic side effects. And of course, even though I just got done saying, and we're going to talk a little bit about the totality of the kinds of pediatric brain cancers that we are addressing, DIPG really, unfortunately, is very visible because of how difficult that was. And no shocker to the rest of us that we really need to find out new ways to do things. I think immunotherapy really, that has been something that I've been very interested in, and many others have as well. CAR T cells fall in this place. The TAT cells that we're talking about today, checkpoint inhibitors, vaccines, oncolytic virus immunotherapy, lots of strategies to try the kid's own immune system to fight cancer. There's a reason for that, or look at any of the real major innovations that have happened in cancer, adults and pediatrics over the last 15 years. It's really hinged pretty heavily on immunotherapy. So even though it was a sort of a theory that people could be cured of their cancer, or they could cure themselves of their own cancer, it had been really difficult to figure out ways to do that until about a decade or two ago. And suddenly many kinds of cancers that, much like DAPG, were incurable, became curable in large swaths of patients. And so that's really where we were focusing on here. As you guys well know, again, CAR T cells, T cells, these are sort of the soldiers of the immune system that, if they can be trained correctly, if they could be grown appropriately, should be able to attack cancer, whether that's in the bone marrow like leukemia, or for our children in the brain. Now, there's a difference between CAR T cells and tumor associated antigen T cells, or TAT cells. There are lots of different subtle differences, but maybe I'll highlight a couple that made us interested in the TAT cell approach. Much like CAR T cells, these products are made from the patient's own T cells, and we don't yet exactly know which patient's immune system is really ready to fight their cancer and which isn't. But the idea for here is, and we'll talk about in a little bit of how we generate these. The idea of T cell is that T cell, if exposed to its target, usually infection, should duplicate tremendously, hone in very specifically, and then turn off when their job is done and the infection is killed, or in this case, when the cancer is completed. So TAT cells, they have a couple of nice advantages, I think. One is they're a little bit simpler and hence a little bit less logistically challenging to make than an engineered T cell like a CAR T cell. For this trial, we also didn't want to just target one target, so we used three separate antigens or targets. You see those listed on the left: WT1, PRAME, and survivin. These are actually normal antigens or normal targets, but they tend to not be expressed in normal cells once you become, once you're born, essentially. But in situations when cancer has deregulated cell division, suddenly a lot of these antigens become presented again. And so that's what we sort of saw across pediatric brain cancers, that a lot of these got presented on a lot of these cancers. The idea still here is you're going to pull out those immune cells, those sort of baby immune cells from the patient, take them outside, train them to attack, in this case, these three particular antigens, grow them. So you start off with like a small army, and then you expand it to many, many, many more soldier cells, soldier T cells, all hopefully primed to attack WT1, PRAME, and survivin, and then you'd infuse those back in. I think many of you might be familiar with how CAR T cells have sort of evolved into most programs infusing those directly into the brain space. We felt like that would miss the opportunity to engage the patient's own immune system in a really direct way. And so our T cells were infused into the blood, not into the brain. It's a clinical trial, and so like any clinical trial, you have to design them with really strict endpoints. This was a safety endpoint. No one had ever given these kinds of multi-targeted T cells, TAT cells, into children with brain cancer before. And we wanted to see how much would be enough for these. There's a little bit of an intuitive sense that more is better when it comes to immunotherapy somewhat. And so we escalated the dose for the patients as we kind of moved through the study. So this did fit into the standard sort of phase 1 dose escalation paradigm. That first stratum is probably the one that really the Musella Foundation and Here Starts Now and many others really focused on, which is in DIPG. So this is for newly diagnosed kids who had diffuse intrinsic pontine glioma after radiation therapy. And then the second two arms are really for the rest of the patients whose tumors had come back and in that context didn't have ready cure. And was really pretty open with any of those pieces. And a small group of patients also received chemotherapy trying to prime those T cells in addition. And we actually have much higher dose levels or T cell numbers than I think a lot of the CAR programs had, in part because we're giving these into the periphery. And so we acknowledge that only a proportion of those are likely to penetrate into the brain space. So we went from 20 million cells per meter squared, so depending on how big the kid was who got these cells, to 40 million, and we got to 80 million. That was our highest dose level, and as we'll talk about a little bit later, seemed to be relatively well tolerated. Like I said, we actually had a very wide swath of patients in terms of who we thought could be eligible, although almost everybody was a child or a young adult. We wanted those children and young adults to, feel pretty good before getting this sort of risky, but hopefully cutting edge kind of therapy. And then we really said that almost any kid who didn't have a real curative option would be eligible with a brain cancer. We learned a couple things, and we're moving pretty quickly through what was actually an 8-year trial and lots of finances and lots of different people working on these. The first is that we found that the T cells tended to be pretty safe. What does that mean? I think that it meant that we gave the T cells and most children didn't have very many side effects that we could detect. That wasn't true for everybody. There were a couple of patients that had more intense side effects, and one child with DIPG, while we thought that the unfortunately lost his life after getting infused with the T cells, thought it was most likely related to the tumor, but we wanted to be very upfront and open on objective, and so called it possibly related to the TAT cells. And then one other patient who was not a DIPG patient, but had a different kind of tumor where we felt like that tumor swelled and gave some potential side effects really in the context of that tumor swelling. Maybe something that was required for response and maybe something that was just a side effect. So overall, this approach of many millions of cells being infused peripherally into these children, all of those cells, once their own cells now reprogrammed to attack three different targets, seem to be pretty safe, meaning most kids did pretty well without many major toxicities, although we definitely wanted to highlight those that were a little different. And these are what we call the swimmer's plots. That red vertical line is that 11th month mark. I know that that mark is a mark that we sometimes think about as, unfortunately, a time when a lot of the kids with DIPG really lose that battle. That you could, I think, see that in many situations with many trials, that red line is probably shifting a little bit to the right. But nonetheless, when compared to historical controls, these are all of the children who got with DIPG who received these TAT cells. And at least we were encouraged to see that many of these children seem to be doing better longer than they had on many other trials, and certainly with standard of care with radiation alone. None of these, except for that one child with the green arrow, I think, who we're still working with today, wasn't enough for any of the other patients, and I think will not be enough for any. So really, we are viewing this as hopefully a stepping stone. Maybe this shifted the curve a little bit. Maybe this will be a good component for a better therapy in the future, and because it did seem like that this seemed to have a positive effect for kids with DIPG, just nowhere close to enough. I think this was a little bit more intriguing for us too. This is a hodgepodge of kids, all with terrible cancers, all that have come back, many of which had spread, many of which had gotten multiple kinds of ineffective therapy before. If you just kind of look at the different colors, those green arrows represent children who are still alive. And if you look at that sort of bottom x-axis, those kids are hitting 4 and 5 years past when they finished treatment, which is really encouraging, I think. But I really like those four colors, the blues and the yellows right there. Those are patients. Those are 3 patients who don't seem to have any tumor that's growing again. Those red bar marks, and this is a table or an image straight from the paper. The red parts are patients who are still alive, which is a win, but whose tumor we think has actually started to grow again. Obviously, a real problem. But those 2 blues and that yellow, those kids are now 3 to 5 years out, and should not have had, based on what we know about their cancers, disease that had stayed away for this long. And so we're really excited that maybe this approach really helped them. Why did it maybe help those kids and not everybody? And why did the kids with the IPG not also have a bunch of blues or a bunch of yellows and a bunch of green arrows? That's the purpose of us trying to move forward with the next step, I think. So I didn't talk a lot about the immunocorrelates. That was a pretty dense part of the paper and a big part of this project. Those immunocorrelates are critical because, as you can see, most patients were not cured of disease, although many of them seem to do a little bit better than we would have expected for them to do. And it's really on us to figure out why they weren't cured. If this therapy is something that can be helpful, we have to figure out why it's helpful, why it's not helpful, who it's helpful in. And that can only be learned through really intensive, and as you guys know, very expensive, not only monetarily, but expensive in blood draws or CSF taps, and lots of time in the lab by a lot of people. That's the only way that we could figure out how to make those better. Overall, there were a lot of positive things. One is we definitely want to have a treatment that causes a lot of problems, although obviously we might accept that if it really cured a bunch of kids as well. But the TAT cells, these multi-antigen targeting T cells that we tested here, got up to 80 million cells per meter squared per infusion. And in general, most children did really, really well with that. We think that it's possible that some of those different colored bars, those kids tended to have a little bit smaller volumes of cancer. It's relevant to mention that that's, of course, none of the DIPG space, where it's very difficult to render a cancer very small in those children. These kids could have had some surgeries, could have had some other pieces previously that just sort of gave them a smaller tumor burden. But we think that maybe that's an indicator of a place, and it kind of makes sense if you have a big army on one hand and a very small defensive enemy on the other, then maybe that's a path forward to efficacy. And we are excited about this. So we are really trying to push these things to the next step, building them to be better, tweaking them here and there, and trying to expand those into other children as well.
All right. Well, thank you very much, Dr. Hwang, for giving us a very detailed analysis of this and help to understand this. I know we have some questions. We have many different chats, many different places that you can go ahead and put in questions. We're going to try to monitor those and try to go through some of those. I do have a couple of initial ones, if I may, while we're going through those. One thing that you mentioned is that this is different than a standard kind of CAR T strategy. And I'm guessing it's more of just it's not this very involved engineering process effectively, and it's using more of the T cells that are resident there. Do we feel that that helps to maybe lower some of the side effect elements that we may see with CAR T in general, or is it that's not the reason for it?
Yeah, Keith, that's a really great question. I will answer that question, but I want to just kind of reflect on that. I talked about safety. I talked about tolerability. The dream would be to have a cure that doesn't cause any side effects, obviously. I honestly think that we are worried very much as a community on the side and the researcher sides of doing no harm. But when we're fighting these kinds of cancers, DIPG, DMG, cancers that have come back, really the goal is to find something that works. And if it takes a lot of work to get through there, to be quite honest, if I could have seen many more blue and yellow kids with lots of green arrows and they had all gotten sick to get there, I think that we would have accepted that as an immense victory. So the safety is important. It's an important question because it allows us to do combinations. It allows us to push the doses. We haven't, in general, seen a lot of toxicity, except in a couple, but not all, of the CAR T cells for DIPG. And I think that's for several reasons. Yes, I think that the way that these T cells were generated probably sort of lends to their greater tolerability. I hope that that doesn't mean that they're in any way less efficacious, and at least our data seem to be put against other CAR T cell programs where the survivals and the responses seem to be also relatively similar. It's also true that I think that by targeting multiple antigens, we were hoping that we were going to be able to take advantage or to overcome one of the big obstacles, which is that all the kinds of cancer cells, whether they're DIPG or others, within a given tumor are very different. They have different sort of susceptibility vulnerabilities, and we were trying to hit multiple ones of those. Now, doesn't mean CAR T cells can't do that. There are CAR T cell programs now that are adding 2 or even 3 or 4 antigens to the same T cell. There are other CAR T cell trials where they're just mixing a bunch of CAR T cells that target a bunch of different antigens as well. So that's not solely an advantage of the TAT cells. I will say I want to also be very open-eyed and fair and objective. So in making one of the differences when we make our T cells is an engineered cell has a greater ability to focus every single T cell on the antigen or the target that they want. So the purity of a CAR T cell product to attack GD2 or B7H3 or GPC2 or anything else, that purity is a lot higher. And intuitively, that might be an advantage of the CAR T cell program. But it was fascinating to us that despite the fact that we had a heterogeneous group of, this is kind of in the small print, but I want to again be very fair about what we should have. We had products that attacked those targets just like we wanted to. We had products that didn't do a wonderful job doing that. They had smaller populations of antigen-specific T cells to those 3 antigens. But they had a lot of activated sort of undirected T cells also. And there's been literature, I think, in the last several years to suggest that the specificity of the T cell product, it's unclear how much of an advantage that is, or whether that's the right path forward either, or whether it's a mix of cells. So at the end of it, we were extremely happy with those kids who responded to their T cells, obviously very driven by those, unfortunately, the most children who were not cured by this approach. But I think that there's still a lot to be learned about T cell physiology and which way is best. Again, we also mentioned systemic administration versus CNS administration. I don't know what the best is. You'll hear we debate in the small back rooms all the time. which is best. And now I think that there's some trials that are actually leveraging both, which would be extremely fascinating.
Can you talk a little bit about the dosages? Did the higher dose group do better than lower dose group? And how many infusion doses were you able to make on average per patient?
Yeah, that's a really important point also. So in the number of doses, so we did not freeze patients, meaning we didn't put a patient on sort of a pseudo dialysis, which gets you a lot of starting material, which really a lot of the CAR T cell programs have to do to generate the numbers that they need. We were able to generate 2 to 4 doses per patient. That is probably not enough. Now, I say that despite the fact that some of the kids with recurrent high-grade brain cancers seem to be cured at this point, and what enough is unclear as well. At the end of the day, we can generate tons of T cells and just keep giving them and hope that they're constantly attacking the tumor. The dream is not to do that. The dream is to somehow make the patient's own immune system wake up, because the elegance, the flexibility, the adaptability of a patient's immune system far outstrips these engineered things that we can do ex vivo or in the lab. So I don't really know. We weren't able to make as many doses as maybe we hoped that we would. I'm unclear as to how many doses that is, like how many doses you really need to, if it's going to work, that this is going to work. One last thing I'll say, in leukemia, where CAR T cell has really, B cell leukemia, where it has really clearly been curative for kids who did not have cure, it's a single dose for those patients. So it's not multiple doses. And I wonder if it's because it's systemically administered and is able to do some things, in addition to all the other barriers that we talk about for immunotherapy.
No, and that's why this trial is happening. Can you speak a little bit about lymphodepletion and whether or not this was used?
Yeah, we did that in 4 patients. It was kind of a tack on, and we tried to do it to answer a little bit of the question. This is another one of those debates. You have some pretty, I think, fierce advocates for lymphodepletion doesn't help, and others who think lymphodepletion is very important. I'm a little bit more on the lymphodepletion, I think, is helpful. I think it's particularly helpful if you're giving systemic adoptive cellular therapy, because it does, I think, sort of reset the immune context and landscape. It may not be as necessary or as differentiating when you do intra-amio or direct to the CSF delivery, although I think that there's some good data to suggest that lymphodepletion can help reset those and make it more favorable too. So the short answer is did not use it in most because we ran out of money, but that was a question that I think it remains outstanding for the entire community. And I do think lymphodepletion deserves to be strongly considered with these kinds of therapies.
It would, I mean, obviously the thought process is that this is not only a treatment, but maybe it's something that can be used in combination with other types of therapies. Do you see that this has a particular strength in that category of putting it with other types of compounds like a Midaso or something like that?
Yeah. So we haven't tested it with targeted therapies or medicines
Or even
like
vaccines, sorry.
one or vaccines.
Yeah.
Unfortunately, I think as everybody knows, there's actually a ton of really great ideas, great trials, and we have to pick what we think is the most feasible or the most exciting, depending on our resources. So we have 2 trials moving forward that sort of backbone onto sort of build on what this was, but only one for DIPG. So Brian Rood, who's one of our lab and clinical neuro-oncologists at Children's National, has actually developed something. He's developed a pipeline which looks at proteins and DNA and RNA in a given tumor at the moment that you use it. And he is using that information with a very complex sort of black boxy to me computer prediction algorithm to figure out up to 20 targets that are the most relevant, most targetable, and most pertinent in that patient's tumor. That trial is targeting children or infants with an aggressive tumor that's not DIPG, or in children and young adults who have a tumor called ependymoma. When that comes back, it tends to be incurable, although it's a lot slower of a process and a deterioration than it is for DIPG. And so we are using the same backbone, but instead of these, he's using a de novo, like just the targets that are given for a patient T cells. For DIPG, Lutza Salante, another investigator at our institution, and Lindsay Kilburn, somebody who I think most people have heard of, another wonderful neuro-oncologist and clinical trialist. They are really leading a trial called LIFT, which is just This is combining the exact same TAT cells, the ones that we just used in Remind, purely for children with DIPG, at first at sort of at same sort of cadence after radiation, but in combination with low frequency ultrasound. That's something else that we've been really interested and excited in for two reasons. One, delivering these T cells peripherally, I think, gives you the advantage of potentially revving up the patient's own systemic immune system. Delivering these T cells peripherally gives you the disadvantage of having only a portion of those cells being able to make their way past that dreaded blood-brain barrier. And so the low-frequency ultrasound device, which is a very expensive machine, uses ultrasound and little tiny air bubbles to sort of temporarily open that blood-brain barrier wherever you're targeting. And so DIPGs are particularly well suited to that. The idea is to use the ultrasound, open the barriers, give the T cells, and hopefully you see a lot more sort of intrusion. So I think that we are just prepping to test this on our first patient. It'll be a little bit slow at first, just so we can be careful a little bit later this month or in early September.
Within the DIPG patients, was there any differentiation of H3K27 mutations within that?
Yeah, that's a great question. Not because H3K27 was our target, but because the immune milieu in those patients are different between H3.1s, H3.3s, and wild types. So we didn't actually require, this was a phase 1 study, so we didn't actually require biopsy. So a lot of these kids were ones who came to us and they had not been biopsied at their home institution. And actually, in that context, we're grateful to have something that made sense and could be innovative, but didn't require tissue. More information is always better than less information. So I do wish we had that information, but we don't know.
Okay. And then, one last specific question with that is, you mentioned obviously the factor of radiation. Was there any differentiation of reirradiation within these patients and some of their timelines?
Yeah, about a third. So that's something that's a really great question, because I think that the risk we run is saying like, oh my gosh, look at our brand new thing. It worked really well. And really what we're really seeing based on this overall survival is just the effect of radiations again, and maybe even again after that. So about a third or two-thirds of our patients did not receive re-irradiation. About a third of the patients did receive re-irradiation. I think that if you look closely, one of the longest surviving patients, two of them actually, who are at 2 and 3 year survival, did not receive re-irradiation, and one of them could still receive it but has not received it yet. If you parse it between the others, I think that to me, what it looks like in its very small numbers is that the re-irradiation didn't have extra benefit because of the T cells, but it gave a similar benefit, which, as we know, is too short, but is a real few months extra with every time you re-irradiate. So it didn't seem to be synergistic. It just seemed to still give you a little benefit, like it would have if you had never received the T cells.
Is there any difference in cost between this and CAR T? How's
Yeah,
it?
this approach is much less expensive for a lot of different reasons. From a manufacturing standpoint, they are still in sort of GMP cell therapy facilities. That part is, I think, but it's easier to scale this kind of approach because you don't need a lot of the components to make the CAR T cells for this component. You need different ones. It's about half the cost of the CAR T cell on a per patient perspective. I think that's a really important question from how you sort of deploy this kind of technology. At the end of the day, I don't really care how expensive the CAR T cell is if it works. Although it is helpful for us to use these T cells in other trials for them to be a little bit cheaper.
I know one of the questions was, what's the next step for this? But I think you may have answered that all the way through this, talking about Kilburn study and Rude studies. But what other things are we missing that are on the horizon for where this goes next?
Yeah, I don't think it's this particular product in particular, but Keith, as you and Alan, many of you on the call knows, there are a lot of really smart people who are pulling from all the technologies at all the single institutions, trying to figure out best how they combine in a way that optimizes each one of them. I think that many of us conclusion that we need a bunch of little pieces that all help a little bit, and that's going to get us somewhere. That happens to be the model. The funny thing is, it's not funny, haha, but funny, odd. The funny thing is that in the 40s and 50s, brain cancer was more survivable than leukemia was. And they basically found little bits here and there and threw them all together. And then now they have like a 95% cure rate for standard risk B cell leukemia. And that's a little bit of the roadmap that I think that we're trying to figure out. But you got to put all these like really esoteric pieces together. And so these T cells, many different kinds of CAR T cells, ONC201, targeted therapy, LIFU or low frequency ultrasound, different kinds of radiation, virus, they're all being sort of put together in different components by a lot of collaborators. So for these T cells, this is in the mix. They're in the mix as well, but we'll just have to see what we are able to move forward.
Well, and you take us into the last question, and that is, what can we do to help within this community, within the foundations, even families that are part of this? How can we help support that and help to allow that next step or that incremental progress that you were speaking of?
Yeah, I should ask you this, Keith, because you will know that a lot of the trials that we've tried to run would not have happened without support from places like you guys and many other people. You saw that acknowledgments page where there were 6 different family foundations that had really contributed, and including to some federal organizations as well. So I hate to start with money, but that's always a piece of it. But it's only a piece of it. I think that also one of the things that I've really um, adhered to in this group is this passion that intersects the families and the scientists. I haven't ever really seen anything like that. So continuing to invest in those visions, bringing people together, talking through things, sort of empowering them to run small things and big things, and really forcing them to. I mean, we no longer have to be forced, but at the beginning it was harder. But forcing us to try to like think about together whose technologies worked best together in what ways. Those are some really exciting, I think, pieces. The other part is once trials do come up, there happen to be uh, a ton of trials for children with diffuse midline. And so trying to facilitate families to know about these trials, to access the trials, to be supported, because it's a tremendous burden. When you have this incredibly sick kid, or a kid who should be sick but looks great, but you know what's happening, but to be able to uproot and go places that have some of these trials, I think that that's another piece that would be very helpful to, I think, both the families and to the community to try to move these research points together.
Well, thank you, Dr. Wong, for walking us through this and talking about some of these big ideas and the next steps that we go with that. If folks would like to learn more information about this, I'm assuming they can probably visit your site at
Oh, just email me. I mean,
just
I think that might, yeah. You happen to have my cell phone also, but either one's fine.
or certainly you can reach out to the Musella Foundation, who's producing this webinar. You can also reach out to us through the Cure Starts Now or the DIPG Collaborative, and we'll be happy to help make some connections or try to answer as best we can. But thank you very much for joining us, Dr. Wong.
Thank
So.
you, Keith. Thank you, Al.
And
Thank
thank
you.
you again to the Musella Foundation, Al, Vanessa, for putting this on and having the idea to really focus in on this. As Dr. Wong talked about, it's about putting these ideas out there and forming those next building blocks. And so we're tremendously appreciative of these types of spotlight opportunities that are out there as well. So, Al, any final words?
I hate to say, but I just want to clarify one thing. You skipped over this answer. Were those people who did better at the higher dose range or the lower dose range? Were those
Oh,
people?
thank you, Al. I did skip that answer. That is fascinating because, I just got done saying that it looked like if you had less disease, that you did better. And that would imply that the ratio of attack cells versus defending, you should vastly favor the attack cells, which would then imply that the 80 million cells per dose would be better. But it didn't turn out to be that way. We had a kid who got the lowest dose level. That was one of our longest survivors. We had one of those people who appear to be cured Two of them were in the middle dose level, and one of them was at the highest dose level. It's one of those, I think part of it is statistics, is maybe if you did this in 100 children, you would see more alignment and reflection of truth. But it is complicated. The kinds of cancers, how they work, the kinds of T cells that we were able to generate for them, what they had received before. There are a lot of different variables. So the short answer, Al, you've already heard. I like to start with a complex answer, but I'll hone back in. It did not seem to totally matter. There was a suggestion that the higher dose did a little bit better, but we'll try to learn more about that in the next studies.
Actually, one more quick question.
Yep.
You picked these three targets. Do you still feel that those are the three best targets, or would you start experimenting with adding other targets?
Yeah. So part of that, I think, is going into Dr. Rood's sort of project where he's now leaving the idea of a standardized targeting system. When you do it for each patient, it takes more time and it's more expensive per kid. But in some ways, you can think about it as more relevant for that particular patient. And so I really like that approach. On the other hand, Dr. Kilburn and Dr. Salante's trial is using these exact same three targets. And part of that is because they're more excited about the combination. And from a regulatory standpoint, having now rigorously tested this particular product is something that's very helpful to use that benchmark and then move forward with the next. My best guess is that I bet the truth lies somewhere in between. I bet that there are specific targets that are probably mostly individualized that are particularly helpful more helpful than others. But I also am starting to think that some of our belief that we can effectively target these things, and to do so, we just have to say you can only target this, and it's only these two things or whatever. I'm wondering if we're giving ourselves too much credit, and ultimately, just by infusing even some very activated immune cells that can adapt, that would be a portion of the answer that might make things more effective. Again, don't know the answer, but it's a good question if I'm like, I don't really know.
And
All
there
right,
and
very well.
thereby you set up the next entire webinar talking about the modeling.
Yeah.
Have a good night.
Uh,
Oh,
all right. Thank you, everybody.
all
Have a
right.
nice
Okay.
evening. Thank you, Dr. Wong.
In that case.